Hello, my name is Aideen Culhan and I am delighted to introduce Dr. Elena Fertig today. She's an Associate Professor of Oncology, an Assistant Director of the Research Program in Quantitative Sciences at an Associate Director of the Convergent Institute at St. Johns Hopkins University, Sidney Kimmel Comprehensive Cancer Center. She's a mathematician with previous experience in weather prediction, and she's applied those skills to advancing quantitative oncology. She is highly published with incredible science, and she is also somebody that I have worked closely with during the past year when we organized a BEARS meeting on multi-omics data integration. So it gives me great pleasure to introduce Elena today.
Great.
I would like to say two announcements. And the first thing is, please put your questions into the Q&A section, not into the chat. And secondly, on Elena's first slide, you can see the website for the DFHCC, where you can find the recording of this talk and previous talks, and you can also see which talks will be occurring in the future. Thank you. Welcome, Elena.
Wonderful. Thank you so much, Aideen, for your kind introduction, and to Dana Farber for hosting me at this talk. So I look forward to giving this talk and to connecting more in the Q&A as well. So I'm going to talk today about work that I've been doing sort of spanning data science and multi-omics with state-of-the-art high-throughput measurement technologies to try to understand therapeutic response and resistance in cancer, going across sort of different systems. And I think one of the things that we think about in cancer is that ideally we'd like to live in this world where you look at high-throughput genomics data, you apply some sort of black box machine learning algorithm, and then figure out which therapy to give people. And the challenge with that is that tumors are incredibly complicated systems, and they don't necessarily obey the simple rules that we would ideally like to see in making therapeutic decisions. And so some of the features that I've been working over the years to understand are what are the roles that different sort of features play in terms of mediating therapeutic response and resistance that might impact those decisions. So one of those critical components, for example, is time, where you can have cancer cells that are initially sensitive to a therapy that can then develop resistance either due to uncovering of intrinsic resistance mechanisms in the population or development of new mechanisms through evolutionary processes. Understanding the role of the cellular composition and how different cell types in the tumor and the microenvironment can mediate therapeutic response. And finally, the relative spatial distribution of those different components. And what's been very exciting to see over the last few years especially, but over the course of my career, has been how the measurement technologies have really evolved along with sort of our understanding of these things in order to elucidate each one of these different components of therapeutic response and resistance in cancer. So I'm going to walk through that today. So the first variable that I'll focus on is how time is playing a critical role in therapeutic resistance. And so sort of going back to the history of targeted therapies in cancer, there was an idea that if you understood the dominant driver molecular alteration in a cancer, you could give a therapy that would target that mechanism. And so that's shown in these little sort of blue cells in my cartoon. And then what would happen is that would really reduce the growth of those cancer cells and reduce the tumor. Unfortunately, there are cells within that population that are also intrinsically resistant. So when you reduce the population of sensitive cells with that sort of therapy, targeted therapy, those sorts of cells that were already there and resistant can start to outgrow. And there's also secondary resistance mechanisms, which is that the cancer cells can sense the therapy and evolve and develop new resistance mechanisms that will also block the growth of the original targeted therapeutic. So there are some cancers, for example, colorectal cancer, where this progression of resistance is very well established and known through mutations that occur within those cells that are compensatory for the pathways that are being blocked. For example, in the case of anti-EGFR therapies. However, there are other cancers like head and neck cancer, where you have 90% overexpression of EGFR and an association of EGFR with survival that would lead us to believe these would be very effective therapies for this disease. But still, patients develop resistance almost universally within a year or two of being given anti-EGFR therapy. And in spite of this pervasive resistance, there hasn't been sort of a canonical path towards mutations towards resistance in head and neck that has been established in the case of colorectal cancer. So the question is, what's going on, and how do we delineate these intrinsic resistance mechanisms from the secondary resistance mechanisms to determine when we should be giving a therapy and how we should be modifying it? So in order to do that, working in collaboration with Christine Chung, what my lab did is develop a time course protocol to sample cancer cells as they were acquiring resistance to therapy. So what we did with Dr. Chung's lab, and this was in the era before single cell data sets, so using bulk data, what we did is we took cells, exposed them to an anti-EGFR therapy. So these cells were initially sensitive to anti-EGFR inhibitor cetuximab, as is shown in the cell proliferation graph on the bottom and the Matrigel assay on the bottom. And what we did is expose cells to cetuximab every single week and collect them for gene expression profiling, which will be the focus of my talk, as well as DNA methylation profiling every single week as cells were treated with therapy. And over time, what happens is with cetuximab treatment, consistent with our expectation of resistance, cells start to outgrow and actually gain a proliferative advantage when treated with cetuximab relative to the original untreated cells. And as well, it's not only that this is sort of this proliferative advantage, but the resistance phenotype is also stable when you subject these later generations of cells to a colony formation assay. What's observed is that the colonies formed are also resistant and larger under cetuximab treatment. So what we would have expected and hoped is to see these nice, smooth gene expression changes reflective of the proliferation alterations associated with resistance to give us drivers. If we do standard clustering analysis of this time course gene expression data, what we predominantly see is a difference of treatment with cetuximab relative to PBS controls. So the dominant signal that we see is a change due to presence of therapy rather than the smooth dynamic changes that we would expect to see with resistance. So the question becomes then, how do we tease apart this time course signal to really not only experimentally get at intrinsic and acquired resistance mechanisms, but also get at that computationally? And so to do that, my group has leveraged matrix factorization. Again, sort of we've started looking at this more generally in collaboration with Aideen, but also some specific methods in my group in order to say not only what are sort of the features that distinguish this, but what are the types of patterns that are occurring in the data? And can we learn directly from exploring patterns within the data, what are the features that are associated with resistance? So I'm going to step away from this previous slide where I showed you my heat map of gene expression data into my nice little cartoon world in which I have only 4 genes. So it's very simple to understand. And what's going on is I imagine that the set of samples, so the y-axis for each of these 4 represents their relative expression level. And the x-axis, I can imagine, represents one of the samples. So for example, the samples on the left, I could say, are representative of time course changes in the treated, and on the right, control. And what you can appreciate by eye, looking at this 4-gene cartoon, is that there's clearly one set of features that are going up and down in the first set of time points related to, for example, this description of treatment, and one set of genes that are going up and down in the second set. And every single gene is then a combination of those processes. So for example, gene 1 is only going up and down in the second set of samples. Gene 2 is only going up and down in the first. Gene 3 goes up and down in both, so that's combining these two processes. And gene 4 is going up and down, it's going up and down in the first, and then up and down twice as high in the second. And so this sort of feature of what's going on relating these to patterns in gene usage is represented very naturally in the mathematics of matrix factorization that's trying to break this down into the constituent parts. And our particular approach, called COGAPS, Coordinated Gene Activity and Pattern Sets, is a variant of non-negative matrix factorization. So it sort of enforces this additive style model that I've illustrated here. And it also adds additional constraints in order to model the sparsity of biological systems using a prior distribution. I'll be happy, I'm not going to focus much on that today, but I'd be happy to talk in the Q&A about that if people have any questions. So I'm going to focus today largely instead on how do we apply this and how do we use this to interpret data in the context of therapeutic resistance. So if I go back to my original gene expression matrix, I can see in the clustering I'm getting out a difference between treatment and control. And now I want to see, are there any sort of dynamic patterns that we're seeing in the data? And so what we do is we apply COGAPS to this data set. And when we do that, in this particular case, we observe roughly 3 patterns in the data. And I see a question in the Q&A about how do you identify the inner dimension when you apply non-negative matrix factorization. So what we did in this particular analysis, and what we've tended to do in practice, is we apply COGAPS for a bunch of different data, a bunch of different dimensionalities, and we observe where the system shows stable patterns versus where it starts breaking down. And when we do that in this particular case, we observed 5 patterns. 2 of them were sort of focused on technical variation in the data, and 3 of them were associated with very clear therapeutic response patterns. So one, for example, was enriched, the first one, was enriched in cetuximab treatment relative to PBS controls across the board. One was slowly increasing with resistance, and one was slowly decreasing with resistance. So this is nice because it gives us these sort of features and this confidence that by virtue of using these approaches, we're able to distinguish which processes are slowly increasing over time, resulting from the acquired resistance, and which ones are really coming in the data just because of the fact that you treated cells, and like we're seeing in the clustering, that introduces a stress response into the system. So that's very nice. And then my collaborators always come back to me and go, okay, Alana, so you found these patterns that are multidimensional. How do you know these have any biological meaning? So that brings us back to having to look not only at what's going on across time and across treatments, but also what's going on across the genes. And so to do that, one of my postdocs, Genevieve Stein-O'Brien, had a very clever observation that typically when you're applying sort of these feature learning systems, what people do is they look for the genes with the highest weights in the system. What that's going to do, if I look at my cartoon example for here, is really prioritize gene 4, for example. But gene 4 is not what's necessarily biologically most unique or most associated with therapy, because it's occurring in both the set of samples on the left as well as the samples on the right. And so what we do is we select these pattern marker genes to find the genes that are unique to each of these. And when we apply that, what we're going to see is that we see these same sorts of patterns in this very nice, smooth process that we can then visualize in a heat map and relate back to specific genes. And when we apply this particular approach to these data, what we observed was a dominant mechanism was compensatory growth factor signaling through FGFR, which was associated with resistance. So then the next question that comes up is, I said the goal of this analysis was to delineate sensitive and intrinsic and secondary resistance. What I found in the previous analysis was a smooth pattern that was associated with increase with resistance, but I didn't have the ability to delineate intrinsic resistance from secondary resistance. And that's really where I think some of the single cell technologies can come into play, because what they can let us do is look at the transcriptional state of every single cell in the pretreatment population and see how much of these signatures that we're learning in relationship to post-treatment are occurring in the pretreatment. So the ones that would be in the pretreatment population are likely intrinsic resistance, whereas ones that are occurring later would likely be due to evolution. So we focused in this analysis, based on our time course data, on compensatory growth factor receptor signaling through FGFR. And in a previous study, also using this approach, what we had found was a sort of central transcription factor, TFAP2 alpha, which was epigenetically regulated to modulate growth factor receptor compensatory activation. And in other previous studies by myself and others have found that epithelial to mesenchymal transition was also critical in therapeutic resistance. And so we wanted to see how much of these are specific to one specific cell line, how much of these are pre-occurring, and how much of these are adaptive. So we collected single cell data of 3 cell lines, one of which was the same SCC25 cell line that previously we had looked at the time course of resistance in, as well as 2 additional cell lines. And we collected both treatment and control in a very short-term response, so after only 6 days. And we wanted to evaluate how much are we seeing compensatory growth factors through TFAP2 versus how much are we seeing EMT, which we evaluate through Vimentin. And when we look at these 3 different cell lines, what we would observe is considerable heterogeneity. So for example, in SCC1, we observe that the cells are largely TFAP2 alpha, and that occurs both pre and post therapy. So this suggests that these sort of compensatory mechanisms of growth factor receptor activation are inbuilt to the cells, regardless of treatment in this particular cell line. In SCC6, which is another de novo sensitive cell line, we see almost across the board Vimentin positive cells suggestive of epithelial to mesenchymal transition. And in SCC25, the same sensitive cell line that we were looking at in the time course data, we observe co-occurrence between Vimentin and AP2, suggestive that the cells can leverage both of them in resistance. So what this is suggestive of is that a lot of the resistance mechanisms that occur over time in acquired resistance are actually occurring within subsets of the cells in the pretreatment population, suggesting that they're hardwired into the cells and give rise to intrinsic resistance that outgrows naturally in absence of a mutation. So this is a little bit challenging, and it also poses a critical issue in terms of evaluating the heterogeneity, because each of these cell lines is going to use a different mechanism to get to resistance. So it's something that we need to consider when we're evaluating therapy. So in addition to just this temporal aspect of the data, it's also within the tumor, right? So this is all in a petri dish, right? Looking at just cell lines. But in reality, when we look in a tumor, these are complex ecosystems. that are engaging interactions between multiple cells. And so, for example, in the case of cetuximab, which is a monoclonal antibody against EGFR, one of the things that can happen is that NK cells can come in, recognize the antibody binding of those cells, and trigger an immune response that causes the cancer cells to die. So it's historically been thought of with cetuximab that its dominant mechanism of action is through blocking EGFR, but it also has this immune-mediated response. And so my collaborator, Lou Weiner at Georgetown, went and conducted a similar experiment to our time course experiment within bulk data from cells exposed to ADCC to evaluate not only what are the resistance mechanisms to anti-EGFR therapies, but what are the resistance mechanisms specifically from immune attack. So they collected similar time course data in this ADCC system. And what we did is we take the same notion of pattern marker genes in order to find sets of genes that are associated with the patterns with resistance, and now plug them into a gene regulatory inference system of string to build up a regulatory network looking at the processes associated with resistance. And what we find is we find interconnected modules of histone-related genes and interferon gamma, suggesting further epigenetic regulation of interferon gamma as driving this immune-based resistance. So we see compensatory growth factor receptors that are epigenetically regulated in the case of resistance to just anti-EGFR killing, and we see further epigenetic regulation of interferon gamma in the case of the immune killing. So this is suggestive of non-mutational mechanisms of resistance in both anti-EGFR, so sort of signaling, as well as immune modulation. So then the next question becomes, again, how can we tell if this is pre-existing or if, is this new to the population? So David Zahavi in Dr. Weiner's lab further collected cells that were either de novo sensitive to this ADCC, this combo of cetuximab and NK cells that were challenged, meaning they were treated with the NK and anti-EGFR therapy, and cells that had acquired resistance. And Teresa Guo ran a UMAP clustering analysis, and what we had expected to see was a subset of the sensitive cells looking like the resistant cells. What we saw was exactly the opposite. We saw a subset of resistant cells that looked a lot like the sensitive and challenged cells, suggesting that instead of a pre-existing resistance mechanism, what we have is incomplete penetrance of the phenotype of resistance. So we wanted to see if we can go a little bit further in order to delineate the molecular mechanisms that we've associated with resistance into these populations to evaluate whether they're pre-existing. So in order to do that, we developed a sort of compensatory process to our COGAPS pattern detection of transfer learning called PROJECT-R. And what we imagine is that if we're applying these matrix factorization tools, and I see there's a question in the chat about sort of other pattern extraction approaches, which I'll get to at the Q&A. But if you sort of apply any pattern detection algorithm, you imagine that you learn sets of genes that can be thought of as a lens through which you view the data, right? Reflective of different cellular states. So for example, in my first time course, there was one that was associated with increase with resistance, one decrease with resistance, and one treatment. Those become the lenses through which you view the data. Then what you can do is once you have the gene weights that are learned from one of these pattern detection algorithms, you can imagine taking those gene weights and applying a projection, just a sort of simple, a simple basically regression of the gene weights that you've learned to see where do these patterns that you've observed in the data, which cells are they occurring in a new data set. So it allows you to transfer the cellular features that you're learning from one system into other cellular contexts. And it can give you more information than marker genes for these processes because you're sort of carrying forward the whole gene weight signature that you're using. And it allows you to really assess this use in a new target data set. And the system, I'm going to focus today on applications with COGAPs, but the Projectar software for transfer learning is really general for any sort of clustering or unsupervised learning method. So when I apply my COGAPs analysis in this sort of ADCC resistance system, this immune killing resistance system, and apply it to the single cell data from the same system, what I observe is that the sensitive, challenged, and resistant cells all follow a trend where the sensitive express the least amount of this signature and the resistance the most. So it does suggest that by and large, the resistance signature is using this program that we learned from bulk data. And in addition, what we observe, and I've highlighted here, is some rare pre-existing subclones that are occurring within the sensitive population that were completely hidden from the initial analysis with clustering. So this suggests that it's largely a clonal outgrowth in the system that's associated with resistance, as well as existent in the pre-existing population. So this suggests the mechanisms that we're learning from bulk are largely intrinsic resistance mechanisms. The other thing, and this is relatively newer data from Ross and Urbe and again, David Zahavi, is that this ADCC resistance, this immune killing resistance, also is preserved across different cell lines, across different cancer backgrounds. And not only do we observe that, but this same resistance signature that we defined in A431 cells occur within other cell lines. So it does seem to be a more robust feature of resistance to immune attack that's occurring across different contexts. So that brings us, so this is a nice experimental system that lets us evaluate that. But one of the things that's really fortunate with single cell technologies is now we're actually able to go directly into the tumor to look at these cells, right? So in experimental systems, you're kind of limited to which cell types you can look at the interactions between. Whereas within an actual tumor, you have this very complex microenvironment of multiple cell types with different types of states, where, for example, if you look at immunotherapy, then responsive tumors are going to have a large number of T cells where you're going to have antigen presentation on the cancer cells that's able to be recognized by the T cells and killed off. Whereas immune cold tumors are going to have a wide variety of resistance mechanisms, including large predominance of immunosuppressive cells that are going to block that response. And again, with single cell technologies, we're very fortunate that we can now take these large tumor ecosystems and look at them, dissociate them, and then tease apart what the molecular signatures are in all the different cell types to discriminate the molecular changes within each cell type. And it's very fortunate that we can do this without having to use an experimental system, so we can try to start directly evaluating from the tumor samples how things are responding to treatment. And a lot of the focus on these sort of immunotherapy treatments and response have been very much focused around what are the cell types that are there, right? Do you have a larger predominance of T cells, for example, and is that why you're seeing more killing? But Aviv Regev in her review here, really sort of highlighted one of the powers of single cell technologies is that you can go beyond just looking at rare cell subtypes. And also these transcriptional signatures delineate different axes of the cells. So for example, you have continuous phenotypes where, for example, cells can go from regulatory to inflammatory. You have cells that can evolve in their types of sort of progression. You have cells that can change cell state, like cell cycle. And also the spatial position and co-localization is going to impact the types of gene expression signatures you see in each cell. And with single cell data, you're able to delineate these axes, but then the question becomes, how do you analyze them? And again, matrix factorization tools are incredibly powerful to sort of mirror this image that you have these multiple axes and figure out what the interpretable biological processes are from these data. So as an example to see whether they enable us to evaluate the role of therapeutic response in cancer and find axes that go beyond cell types into more continuous cell states and features, what we did is we took single cell data from a mouse sarcoma model that was sensitive to immunotherapy. And so there were control cells treated tumors, control samples with anti-PD-1, samples with anti-CTLA-4, and samples with their combination. So this was a public domain data set provided in the study by Gubin et al. And in their original paper, what they focused on was a lot of standard clustering analysis to define cell types, and did a beautiful job of sort of looking at the role of macrophages and T cells in therapeutic response. What we found when we further apply COGAPs to these data is that we're able to delineate heterogeneity within the NK cells that are associated with this data. When we look at what's enriched in this subset of NK cells, we find that this particular group of NK cells is all enriched in the anti-CTLA-4 treated samples. And moreover, if we look at the marker genes applying that same pattern marker approach that we described, what we observe is that this is associated with NK cell activation. So we're seeing NK cell activation in association with anti-CTLA-4 response, suggested if it's not just a T cell phenomenon, but also a phenomenon associated with NK cells in anti-CTLA-4. So the further question was, okay, this has been shown before in mouse. How robust is the signature that we observe in mice occurring in humans? And is this relevant to the patient response to therapy? So now we go back to our transfer learning approach, and instead of focusing on sort of bulk to single cell technologies like we did in our previous studies, now we focused on looking at mouse to human and seeing how much does this let us evaluate cross species what's going on in therapeutic response. So Emily Davis-Marcisak, who led this analysis, took this Gubin et al. Data and compared it to several human data sets. And I'll focus on one specifically by Sadeh et al., where that focused on immunotherapy response in metastatic melanoma. So again, we're going across disease contexts from preclinical to clinical and looking at this sort of response in relationship to therapy. And what we find is this NK cell activation pattern that we observed in mice is associated with enrichment in responders to anti-CTLA-4 in pretreatment data in humans. So we're observing that this activation of NK cells is necessary for the response in anti-CTLA-4. And so the question is, why hadn't this been found before? What about this wasn't known? And can we sort of go back and validate that this is occurring in human samples? It turns out that NK cells can be a little bit tricky to get flow cytometry working on to sort of evaluate their presence. So what Emily did in collaboration with Alison Fitzgerald and Dr. Wiener's lab is instead apply standard molecular biology of qRT-PCR and Westerns to look across panels of human NK cell lines, as well as human donor NK cells, and observed that in CTLA-4 treatment, that these indeed express CTLA-4 both at an RNA and a protein level. And further imaging, they conjugated a fluorophore to ipilimumab, which is the anti-CTLA-4 inhibitor, And they found that indeed it does bind on the surface of NK cells, supporting a direct role of NK cells in anti-CTLA-4 killing. So as the final step of my talk, so I've sort of talked about different cell type composition that mediates therapeutic response. The final piece of this is it's not only what cells are there, but what are their relative spatial localizations. So in this cartoon, I'm giving 3 cases that would all be resistant to immunotherapy. And this would all occur for different reasons. So on the left, it's that you have this heavy sort of infiltration of immunosuppressive cells within the tumors. In the middle, it's that you have activated T cells that aren't able to infiltrate. And on the right, it's that you have exhausted T cells that are infiltrating. So all of these are different mechanisms of resistance that really need spatial technologies in order to resolve what's going on. And we're very fortunate that spatial molecular technologies are really at the cutting edge of single cell, where we have spatial proteomics, which are really exploding to get very high cellular resolution and somewhat decent protein resolution, probably on the order of 30 to 100 markers, in order to resolve these systems in a way that relies on antibodies in order to target specific cell types. And at the same time, we also have new technologies for spatial transcriptomics, where there's a trade-off between cellular and molecular resolution. And there's technologies coming from companies such as 10x with Visium that are really advancing towards whole transcriptome cell level profiling with challenges and caveats. But it's a very exciting opportunity, I think, in the field where these technologies are really exploding in order to look at these sort of therapeutic mechanisms. So in collaboration with Visium from 10x Genomics, they provided us with a sample data set of an invasive ductal carcinoma tumor. And the Visium technology has H&E in combination with expression of 25,000 sort of whole transcriptome-wide data. And this poses a very exciting opportunity to look at these data. For example, looking at the relative spatial distribution on the right, I've shown for standard marker genes of immune activity, for example, CD8 as a marker of CD8 positive T cells. And sort of see how those relate to the different lesions that we observe within the tumors. So the question is, how much can we then tease apart? Are these lesions using similar molecular signatures? Are they using different molecular signatures? How does that relate to what we would expect to see and the way in which this carcinoma is invading and immune infiltration? So again, this is sort of another single cell data modality. We're going to apply our same matrix factorization approach to see what are the dominant molecular signatures that we observe in this. And when we apply this signature, what we observe is we observe sets of patterns that distinguish each of the different invasive lesions in this tumor. So for example, we observe one set that's associated with the invasive carcinoma, as well as several others, for example, pattern 15 on the top left that's associated with one particular DCIS lesion, pattern 14 on the right, which is another DCIS lesion, whereas pattern 11 is a common molecular signature across the different lesions. And what's interesting is each of these, if we look at the genes that are associated with them, are associated with a different set of hormone receptor signaling pathways, suggesting this is going to be a tumor that's going to be very tricky to treat with a lot of traditional targeted therapeutics because you have this wide range of heterogeneity in the molecular processes it's using across the different lesions. So to summarize sort of over this talk, again, therapeutic response and resistance is really a dynamic process that depends on cellular evolution over time, cellular composition of tumors and their microenvironments, and spatial distribution of cells. Time course multi-omic study designs are really critical to understand intrinsic and adaptive resistance mechanisms. And single cell technologies and emerging spatial molecular technologies are at the cutting edge of these approaches. We've found that COGAP's matrix factorization is robust to this, but I think, I see a question in the chat too about this, about other traditional pattern extraction approaches such as self-organizing maps, I think that really unsupervised learning in the context of these molecular processes with resistance is at early days. And so there's been a lot of work with it to look at it in context of single cell technologies, but really what is the optimal for understanding therapeutic response and resistance mechanisms and reproducibility for the clinic, I think is still an open question that people need to answer. This sort of concept of transfer learning, I think, whether you use COGAPs or some other approach for unsupervised learning, is really robust to identify both pre-existing resistance mechanisms, but also relating preclinical models to patient samples and back again. And I think future work is really critical. This is a very active area of thinking about what are the approaches that do best to figure out what's going on in the tumor cells? What are the approaches that do best to really figure out what's going on in immune cells? And how do we infer the interactions between those two? So this was obviously work that was done in huge collaboration with a number of collaborators, and particularly grateful to members of my lab who did a lot of this work. Emily Davis-Marsasak, who led the NK work, along with Allison Fitzgerald and Dr. Wiener's lab. Luciene Kagahora and Genevieve Stein O'Brien, who led the time course and study of compensatory growth factor signaling in collaboration with Christine Chung's lab, and Melanie Noloff, who's been focusing a lot on the spatial molecular technologies. So thank you.
Thank you, Elena. That was a fabulous talk. We will answer some of the questions. If you have questions, please put them into the Q&A. Elena, so how does Project-R package different from Vision or FAST Project?
I apologize, I have not heard of Vision, so I can't answer that one. In terms of FAST Project, I think the underlying computational framework is similarly. We tried to develop the software very flexibly so that you could put in different like clustering algorithms or different data structures of the low dimensional embeddings that are embedded from bioconductor classes so that it could work across different methodologies very naturally. But I think the underlying math is very similar to FAST Project.
A naive question, how do you transform the normalized RNA-seq expression data that contain negative values such as log2 transformed TMN normalized read counts into the format that's acceptable for NMF?
So we usually do some sort of variance stabilization and log transformation. Typically we add a +1 so we don't end up with any negative values when we're looking at RNA-seq data. But we've also used other cases where you can sort of shift everything up, and because the model is the same, whether you have a constant +1 term, versus separate patterns, you can shift everything up so it has minimum 0 without a problem.
Rafa would like to know, can you say something about the advantage of NMF over something based on SVD?
So a lot of the SVD and PCA approaches are very robust at delineating sources of variation in the data. So they're incredibly useful. What we've found is they tend to mix together different biological signals. And so you get really robust clustering with PCA or SVD, but you're not necessarily getting additive processes that are uniquely associated with one sort of feature versus another. So for example, you might not delineate something, you might, like in my case of the time course data, you might confound therapeutic response with the slow sort of feedback of resistance, whereas those come out as equally weighted processes through NMF.
So there's two questions here. I think one you referred to already, which was the advantages over traditional pattern extraction methods such as self-organizing maps. But there's a second question here comparing to autoencoder-based representation algorithms, pros and cons. So I think a lot of people are interested in comparing NMF approaches to other approaches.
Yeah, and I think that that's a really robust area. I think it's something that, you know, as we talked about in our workshop, we don't have great metrics for evaluating. And so there's a ton of other approaches out there, you know, the study where we sort of benchmark COGAPS and PROJECT-R in the developing mouse retina, what we found was NMF was a little bit more robust at getting both sort of continuous processes related to development as well as cell types. There were some cell types that were better resolved by autoencoders and some better by NMF. So it kind of depended on which cell type you were looking at. So I think the trick as a community is we're going to have to figure out how do we want to validate these, right? Because it's very easy for us to come up with computational metrics to evaluate this. But I think the biological metrics of what are we resolving with these different approaches and what are the sort of gene weights is really an open area. And we think that's where transfer learning, and part of why we think sort of developing the standardized approach that works across methods is really robust, is because then you can say, okay, what does this preserve across other data sets to evaluate the biology that's preserved across them? But I realize that's a less than satisfying answer of like, use this one in this case and this one in this case. I don't have a good answer. And I do think the solution is going to end up being some ensemble across methods. A lot of my group obviously developed COGAPs, so we have a bias towards this NMF approach. But I do think that a lot of the stuff we do in terms of interpretation and visualization and gene extraction really can be abstracted beyond the NMF framework, and is a lot of where we're focusing on a team is going is if you have these low dimensional representations from any system, how do you understand if they're interpretable? How do you relate them back to biomarkers? How do you relate them back to processes? Because we're very concerned with translating them back into clinical or experimental decisions.
I think that's really important. Like even translating from in vitro to clinic has got its issues, and understanding those limitations I think will be vital. And generating benchmark data sets. I'm going to ask a question here myself, which is, if we were to design a benchmarking study to try to evaluate these approaches, what do you think it would look like?
I mean, I think it's the approaches, and then it's the approaches in the clinical context, right? So there's been a lot of work in benchmarking the computation. I think that's been done in developmental biology, which has this huge advantage, right? The systems have to obey in very constrained ways in order to really like survive, right? Because in developmental biology, if the system's not following the sort of linear progression, it's either going to have a vastly different phenotype or typically it's lethal. And so it makes it a very constrained system to study things, which is part of why we sort of, when we went to single cell, we started looking at these in the retina. And the nice thing about some of these developmental systems too, is there's years of experimental data. And so I think, you know, as you and I have talked about, like multi-platform data, maybe where you have sorted cells and very constrained biological systems are going to be very important for benchmarking the algorithms. The challenges, as you well know, and I think a lot of people here can appreciate, is when you get to cancer biology, a lot of those strong assumptions totally break down. The system is completely heterogeneous, and everything that you benchmarked in developmental biology is not nearly as clean. And so I think there we really need systems where we have these sort of multi-cell types, where we have these robust co-culture systems, and where we have therapies that are behaving sort of canonically. And if you look at a lot of the data, like for example, of a lot of, you talked about in vitro studies, a lot of sort of historical in vitro studies that tried to do this, the first thing you look at, if you look at those data sets and do a deep analysis, is that if you look at the therapies that these drugs are claiming to inhibit, you often see an opposite effect due to feedback. And so it makes it very difficult to have this baseline of what's really going on in the biology, what therapeutic perturbations are really doing relative to the expectation. In communities like the Dream Challenge and Links Project have really done an amazing job of curating these for the community. And I think we need more data sets where we have controlled co-culture models to really evaluate this, and evaluate the immune response is going to be tricky.
Definitely. There's a question here, just thinking again about sort of the biological aspect. How do you narrow down on the histone genes that can functionally regulate the interferon gene cluster?
So in that case, what we did is we took the gene signature that we defined. I'm going to sort of try to rush back to that slide if I can. So we had this whole gene signature that we had from the pattern markers. And previously we had looked at it in this heat map-based visualization. And here we had put it into string. So that basically gave us this regulatory information that was based on prior knowledge of of protein-protein interactions. The way we figured out that it was histone coming first and then interferon gamma was by virtue of having time course data, which I don't show much here. But the red dots that I'm showing in this slide are the relative expression levels. And if you go across the multiple time points in this experiment, what we observed was that the histone gene module was high first, and then it decreased in terms of the amount of expression, and then the interferon gamma module followed that in terms of its expression changes. And so I think there's a whole huge role that's really, I think, underappreciated of timing delays between gene modules and between features that are really critical to understand. And I see, like in the Q&A from Serdar, there's a question about fusing multi-omic data to NMF. And there have been a lot of multi-omic approaches developing with NMF, including our own. And what we found is that they work very well if the processes are occurring at the same time point. But in, for example, that time course study that I showed in the beginning of therapeutic resistance, we actually had DNA methylation and gene expression. And we found that the common features that when we applied a full integrated NMF to the approach to that data, it completely didn't work because there were different underlying processes and dynamics in the methylation changes than the expression. And we actually had to analyze them separately in order to understand them together. And that's part of what we think the power of Project R is too, is because you can go across data modalities to see when is this occurring in this system, you know, and how robust is it across different data modalities that is a little bit more challenging in a coupled NMF.
So there's another question here kind of related to that, which is, what about targeting the ability to upregulate, downregulate, or to change models at all by going after the epigenetic machinery? So how do we begin to connect epigenetics, gene expression, and I suppose even onto proteomics?
I think that's going to be an incredibly powerful area, both therapeutically as well as biologically. And there's a lot of work looking at epigenetic inhibitors in context of sort of mediating therapeutic resistance. The challenge is a lot of epigenetic inhibitors are pretty dirty, right? And so they do a lot of things when they're rewiring the system, which is a challenge and a sort of feature. So one of the things that's nice about those is they can modulate these gene changes, and they have been shown to mediate resistance. They also have very interesting immune effects where they can modulate the immune microenvironment and antigen presentation to potentially sensitize systems to immunotherapy. So they're a very robust suite of drugs in order to look at, but because they're so heterogeneous, it can be hard to give them and be certain, for example, that it's going to impact the exact set of genes that you're hoping for it to. So, you know, I think there's a lot of work in sort of the, you know, targeting gene regulation. That's a very open area. And I think it's going to be different if you're in the immune context than if you're in the sort of targeted therapy context.
Yeah, I think the single cell, once we start to look at the chromatin structure and the attack-seq and the single, the RNA expression, and all of these are coming together with single cells. And can you talk maybe about the ability within NMF and COGAP to integrate some of these other data sets that are coming online for single cells?
Absolutely. So we have another paper in nucleic acids research where we looked at this and found sort of similar performance of ATAC-seq, where we were able to delineate different cell types in response using ATAC-seq data with this matrix factorization approach. One of the things that's interesting is then how, sort of related to an earlier question, how do you pre-process the data and what's the right level of summary? So is it most interesting at a motif level of where you're sort of thinking about binding? Is it most interesting at a peak level? What we found was another one of those annoyingly complicated, well, it depends, where, some features were better resolved where you summarize the data at a motif level and some better at a peak level. So we got sort of the most resolution from the data at a peak level in spite of the sparsity using COGAPs, but we still see additional new features that come out at a motif level. And so I think, again, thinking across all these different unsupervised learning approaches is going to be critical to see how these are mediated. Right now the challenge with these technologies is that by and large, you measure different pools of cells with ATAC than RNA. Now that's changing. The technologies are allowing both at the same time. So that's where, right now you sort of have these projection approaches that are really limiting to studying that. But it'll be interesting to see, even when you have them on the exact same cell, I don't think you're going to see the epigenetic changes occurring at exactly the same time as the RNA ones. And I think you're still going to have to think about how do you model time delays and how do you look between systems. It's going to be very interesting.
And how do we even capture time delays when we're actually generating two data types on the same cell?
Yeah.
Yeah. So there was an interesting question here, and I think it's always related to how you put those methods into a workflow and other things you might rely on, which is what other bioinformatics tools that are similar to COGAPs and our projector do you use simultaneously for each analysis to validate the results, or do you have alternative methods to understand the biology? So kind of what are the other tools in the toolbox?
Yeah, so usually we sort of go back and forth. So we'll do, like Rafa was saying, we'll do like an SVD or PCA to kind of see what's going on. Like I showed you in, this data set, we'll do a clustering, and a UMAP visualization. We'll do differential expression analysis. We'll kind of take the whole suite of things and see. And I think probably most of my papers end up using differential expression analysis because my preference is usually to go with the simplest analysis approach when I'm doing something that's, when I take off my algorithm development hat and put on my, biology hat. My approach is to use the simplest analysis approach that elucidates the most biology. And so typically I'll do all three of them and pick the simplest one to explain the story. But, I think, again, I've found that it's very data set specific where features start to pop. And I think, every bioinformatician can appreciate it, going from a gene list to understanding the story and the data, I think takes a lot of back and forth of different analysis approaches until you see what's actually there, and the data sort of shines what's coming out at it. And I haven't found maybe others have other ideas, but I haven't found any substitute for just going through a bunch of approaches and seeing where the signal shines.
Yeah. I have to say I was very excited to see your NK cell work with CTLA4. And where is that going? What's the next step for that project?
So we're trying to figure out the function right now. So we're doing a bunch of co-culture experiments to understand what's going on, as well as collecting some patient data to see, like, is this robust, and sort of doing additional experimental work. But that's very much in, I think, planning stages at this point, just starting up again with the labs reopening. So
completely. I can imagine several people from Dana Farber will want to contact you about that. Is your contact information, if you want to put it up there for people?
Not sure. It's on my website. Let me see if I have it. And so Twitter is always a good way to get through to me. Oops, sorry, I lost my screen. So, you can always get through to me through Twitter at FertigLab. And I don't have my contact here, but it's ejfertig@jhmi.edu if people are interested.
Thanks, Elena. I realize we're getting close to time. There was a lot of questions. People can follow up with Elena directly if there's additional questions. And I'd like to thank you so much for an incredibly exciting talk. Thank you.
Thank you for having me.